So hi, everyone, and thank you for joining us today. This is our monthly meeting, which addresses tomorrow's life science workforce. Today, this is a charrette for Lab of the Future. At this time, I'll turn it over to Michael Welling, who is the co-founder of Westchester Biotech Project.
So, Thank you, Denise. Thank you, and thank you, Joanne, and good morning, everyone. Appreciate you joining us on this somewhat dreary but exciting day. Spring is upon us. It's St. Patrick's Day, the basketball tournament's starting, and feel like the warm weather is not too far away. I've met many of you, some of you not previously. While, as Joanne mentioned, I am the co-founder of the Westchester Biotech Project. They don't always let me out of my closet to speak, and oftentimes I get in the way. But I like to join and say hello and express appreciation to all of you for your time and your contribution to the work that we're doing. And for those of you who have participated with us in the past, you know our work is a collaborative effort. This is not about our vision, our agenda. This is about our effort in supporting all of you and helping achieve and accomplish your goals and your mission. So this is truly collaboration in motion, and we appreciate all of your contributions. As Denise said, this morning's conversation, we are excited to have John Tamborino from Skanska, Anthony Caius from Synthego, and also Sara Francini from Skanska to talk about the Lab of the Future. This is a concept that while it's not a new concept, we didn't invent it. It is a conversation that we have found has been a challenge for so many in the biotech community that are not currently in New York City and Boston, and even they are having this conversation as well. So we welcome all of our speakers and discussants and participating in today, leading this conversation and helping us sort of forward the collective momentum in addressing the challenges that are needed to build out this community for tomorrow. Next slide, please. Obviously, a huge thank you to the folks that are doing all of the legwork and putting these sessions together and these conversations. Carla Romney and Ray Houston, who have both just been with us from day one and have been invaluable both in their contributions and their insight and support of framing these conversations. And a day does not go by that after I thank my wife for all that she does for me, that I don't thank Joanne for all that she does for our organization here. So Joanne Gear, for those of you who don't know, is the spirit, I think, behind all the work that we do here and helping us achieve our success. And while your name's not up there, Denise, I'm going to say a big thank you to Denise. As you all know, she is the one that keeps us focused and moving forward and organized and professional and mature in our discussions, even when we disagree. So thank you to Denise as well. Next slide, please. This is a quote that we have been sort of leaning into as we have crafted our goals and missions and vision and strategies. And again, the Lab of Tomorrow is just no better example of this, that we're not going to accept what today is the end-all be-all solution. We are not going to accept that tomorrow will just solve itself, but we want to be part of the solution, if you will. We don't see ourselves as the sole solution, as evidenced just by all of the contributions you'll see today, but we are proud of the work that we've done in bringing people together and trying to make tomorrow the best version of itself that it can. Next slide, please. How do we do that? And for many of you, you know this, but it has been our mission to focus on the supporting of and celebrating of researchers, data scientists, and engineers, the folks that really are at the heart of any great innovation, any great research company. Without them, all of the talking heads and all of the press releases don't have a lot of bite to them. So we have felt part of our mission has been to highlight them, support them, encourage them, foster the collaboration amongst them, and doing so in what we refer to as borderless initiatives. Our community is global. While we have Westchester in our name, that is more a function of wanting to highlight all the good work that's going on in Westchester, but not that our focus is on only building up Westchester, but connecting Westchester with the rest of the global community that's in this space. And as I said, and we'll continue to say, we are so proud of the work that we've done, but also the recognition of the global community seeing the importance of this work. Next slide, please. The Career Consortium, I'm going to turn this over to Joanne to talk about some of our programs here. But again, I will leave with this final thought. We are so proud of the work that we are doing. We have recognized the impact that so many of these initiatives can have and need to have, but none of it happens without the contributions and participation by all of you. So a big collective thank you to all of you, and I'll turn it over to Joanne.
Thank you, Michael, and thank you for your kind words. I really appreciate it, and I'm having the most fun I've had in my whole career, so it's really quite a treasure. Would you go to the next slide, please? We don't do this alone. We have lots of different organizations and companies that give us support in various different ways. I'm going to highlight one that has made a 2-year commitment to support us and really help us in our growth, which is Montero Language Services. Montero is based in Westchester and in Madrid, and does translation of elite documents such as clinical research documents and intellectual property. And we just had a great relationship with them, and we've done a white paper together and a number of other really interesting outreach efforts to benefit both our organizations. Continue on, please. We have a number of programs coming up every month. We have a couple of things happening. Next Tuesday is our Innovation and Research Conference. This is our annual kumbaya to bring people together to share what they're working on. When we do it in person, it's a full day. This is just a morning online. And our focus this time is on breakthroughs in maternal health. And all the materials are on the website. We have a microbiome program that continues on, and the meeting that we're in today is a monthly meeting, so you can join us each month for a different content. Continue on, please. I'm going to hand it over to Ray Houston, who will tell us a little bit about what he does and the Career Consortium, and then we will start to hear from Skanska. You can take the slides down for a moment. Thank you.
Good morning, everybody. So thank you so much for joining us. As Joanne said, I am Ray Houston. I'm the Dean for the School of Mathematics, Science, and Engineering here in beautiful, rainy Westchester County, Westchester Community College. I have been in this position for a little over 4 years, which actually is about the same amount of time that I've been associated with the Westchester Biotech Project. I was a longtime math faculty member here at the college, and then I stepped into the Dean role, and I've been doing that ever since. Westchester Biotech, it's really special for me because, like I said, it's like the first big outside thing I was asked to do by my boss, actually. She said, hey, here's this meeting I need you to go to, and it's at Fordham, Westchester, and it's at 8 o'clock in the morning, which that was not, I live in New York City, and I'm not a morning person. So I was like, really, this must be a test. But I went to that program, and I've been hooked ever since. It's been it's been wonderful to be a part of it. And I'm always excited to work with Joanne and Michael and the folks, just putting these things together and just bringing a lot of these issues to the forefront, and especially how it connects to my students. Because our students, I think, often have problems connecting with what they're doing in the classroom to where it ends up with in the real world. Like, what am I doing in this classroom that's going to connect me to working on a career, or, okay, after I transfer to a 4-year institution and I complete my bachelor's degree, what do I get to do after that? Those types of things. And so working on the Westchester Biotech Project has allowed me to help shine a light on a lot of these things for students so they can understand, oh, this is what I can do. And another thing, a lot of our students, we have a hard time getting them to leave the college. And part of that is because they love being at WCC, but most of them are Westchester County kids, and they want to stay in Westchester County. So actually providing opportunities for them to, complete their education and start and continue a career in Westchester County is really important to us. And so that's another reason why it's important that, that I feel it's important that I participate in this program. The Career Consortium provides training and networking to prepare companies and educators through cross-institutional collaborations and knowledge sharing. So again, that's really part of the mission of WCC. It really is helping our students understand, okay, this is where we're working on the content knowledge. Now we're going to also build the bridge so you can understand where you can go once you have that, once you have the content knowledge. One of the areas specifically that I've been working on with Carla for 4 years is the Westchester certificate program. It's based on essential skills in the laboratory, and it actually contains 5 areas. So it's laboratory safety, scientific laboratory notebook practice, aseptic technique, sustainability in the lab, and understanding the drug development process. We are very excited that we've already completed a remote version, portion of the laboratory safety portion of the certificate. And in May 17th and 18th, we're going to be having another part of the certificate. We're going to do an aseptic technique and more laboratory safety here at WCC. So stay tuned for that. That's really exciting. And we're hoping down the road we can develop things like micro-credentials and things like that for students to put, so they have badges and things like that to put on their resume to say they've completed the certificate. And then employers can say, oh, they are prepared to work in a laboratory setting. So that's it for me. And I'm going to turn it back over to Joanne so we can get on with the program, because I'm really excited to hear what Sara and John have and Anthony have to say this morning. So again, thank you so much.
Thank you, Ray. So I'm going to introduce Sara Francini in a moment from Skanska, but just to give a little context, so we've done many gatherings, many roundtables, many in person when we were able to do that, number of symposia, and we have really been listening. And the issue that we got over and over again from colleges and from growing companies that there is not enough lab space in Westchester. So I won't go to all the background, happy to go over it with anybody who is interested in the whole backstory. But we decided that we are going to be doing a series of charrettes, looking at different aspects of building labs, helping educate ourselves. We aren't necessarily going to make the whole deal come together, but we can certainly invite the people to the table to find each other between the resources, the needs of companies and universities, and really the whole, taking on the whole idea of how do we create a shared space that can function to help young companies grow and also make a place for internships and apprenticeships and all the other pieces of the puzzle that build the pipeline. So I would like to introduce Sara Francini from Skanska. Go ahead, Sara.
Thank you, Joanne. Good morning, everyone. It's great to meet all of you and to be here today. My name is Sara Francini. I'm the Vice President of Business Development for Skanska's Metro New York region. Skanska has 29 offices around the country. So pleased to be with you here. Just a little more context about Skanska and what we do, particularly in the life sciences. So we are ranked as of 2021 as the top science and tech labs contractor nationally. We've completed more than $17 billion of work in life sciences nationally. We currently have $2 billion in life sciences work in progress. And regionally, we've completed around $7 billion in life sciences work. So some deep expertise there. More than 200 of our employees in the region have life sciences experience. And we're currently involved with projects like the Public Health Lab for the New York EDC. We're working with Cold Spring Harbor Laboratories, biolabs at NYU Langone, and also working with Regeneron at their Sleepy Hollow campus on a fit out. So again, pleased to be with you here today and to get introduced to this great group. We had a little chat in the prep session for this. We've seen in the region an increase in life sciences opportunities, particularly in the past year and a half, both in private industry and in higher education. We do a great deal of higher ed work in the region as well. That trend of increased prioritization in life sciences, in STEM, in health sciences is something we're certainly seeing, both with existing clients and prospects. They seem to consider these investments as a way to really attract and retain top talent and to stand out amongst, competing programs both in the region and nationally. So, with the funding we've seen and the projects moving forward, there is a big focus on sciences, STEM, and life sciences. So it's a very exciting time for us. I'll stop talking now, and I'm just going to introduce my colleague, John Tamborino. So John is our Vice President of Pre-Construction for Skanska's Metro New York region. He brings 43 years of experience in cost estimating and construction management. He is responsible for our pre-construction support services. It includes coordination and supervision of our staff, estimate preparation and value engineering, alternative studies. He has worked on a number of projects across a variety of sectors: life sciences, healthcare, higher education, transportation, industrial, and commercial. And he is also a lead accredited professional and HASHI certified. So thank you for your time. I'm going to hand it over to John. And if you give me one moment here, I will pull up his slides.
Well, thank you, Sarah, for that kind introduction. When you think about 43 years, I'm thinking to myself, I don't look like I'm 43 years in this business. Maybe, I don't know. But I'm very happy here to talk to you today about life sciences, construction, cost considerations. Everybody's talked about already how much there is a need for lab space in Westchester. And we took it upon ourselves maybe a year and a half ago to put together this white paper detailing the cost of lab construction. So, next slide, please. So when we put together the white paper, we understood that there's major cost drivers for lab construction as opposed to, say, office construction. And typically owners and clients and not contractors, but the institutions come to me earlier on in the process and they ask me, well, how much? And usually what I get from them is this, a blank sheet of paper. So I have to understand what the program is and where they want to build it, and what type of lab they want to use. So I put together this white paper and we identified 5 major cost drivers. Obviously, HVAC systems, plumbing, electrical, the lab architecture itself, and also the equipment and casework that's part of the lab construction. Briefly about HVAC system, there's a lot of airflow that goes through labs as opposed to office spaces. And you need to have a lot of air going through a lab space, especially when you get into the higher levels of containment in the lab space itself. Plumbing systems, electrical systems, all intertwined with each other in terms of the lab space program itself. In terms of lab architecture, you talk about the interior flow of a lab space itself, how it's more endemic for the workers and researchers, as opposed to, say, in an office building where the focus is really on the exterior wall of the building. And of course, the last piece here is the equipment and casework. Today, the casework is more movable and flexible, as well as the equipment utilized within the lab space. It has things like biosafety labs and casework and things of that nature, which cost a lot of money. Next slide, please. So as part of the talk, I put together some costs for interiors of lab construction. You see here, the dark blue is on the higher end of this range, and the light blue is on the lower end of the range. And then on the left, in the first columns here, is the lab fit-out cost. On the higher end, $645 versus the $365. Now, I'll preface this by saying that this report was done over a year ago. And as everybody knows, the marketplace is crazy today with inflation and commodities costs, as well as supply chain issues. And I'll get into that a little bit later on in the discussion. The next group of graphs here is for infrastructure costs. Sometimes when you're looking at an existing building, there are other costs involved in infrastructure for that building to bring it up to a level where you can have a lab space. So there's additional costs there that sometimes are forgotten or not known. So on the higher end is around $181, and the lower end is $96. And then the last piece here is enhanced infrastructure, what I call enhanced infrastructure, where an existing building might have to have other things done to it in order to bring it up to code in terms of energy efficiencies, like windows and new roofing. So these are things that you have to consider if you're considering a lab space renovation, build out, or even a new building. Next slide, please. Next. So this slide details lab construction and a Greenfield site, meaning it's not in the middle of New York City. And we also identified certain localities that we do work in, New York being one of them, obviously, Boston, North Carolina, and California. These graphs are done by major building systems that shows the actual cost of each one of those major building systems within the space of a lab construction. So the light blue is the structure, an envelope, that structure. The darker blue is the envelope. The green is the interiors, and that yellow is the MVP cost. The light green is the electrical, and the gray is the lab equipment and casework. So you can see by this graph that the most money you're going to be spending on your space is going to be in the MVP systems, that large yellow vertical stripe. And you can see that most of the other markets are showing the same thing in terms of cost of their space. They're all aligned in those particular marketplaces that has the MVP as the most expensive piece of your work. So it's something to be aware of when you're looking to build a space. And we do work in every one of these locations, as we have a major offices in each one of these places. Next slide. So as opposed to the lab construction, we show here an office construction as a comparison against the labs. Again, the same marketplaces and geographies, New York, Boston, North Carolina, and California. And you can see the same colors represent the same major building systems. And here I will identify that dark blue is the exterior wall component. In each one of the locations that you see here, the exterior wall is the bigger piece of the cost, as opposed to in the lab, the MVPs are the much major cost drivers. So when you're looking at office construction, the expression is really focused on the exterior wall as opposed to the labs. Next slide. So how do we come up with all these numbers? I said before that people asked me earlier on, how much? So what you see before you is the Skanska metrics dashboard. It's our own proprietary database program that provides benchmarking costs for all across the U.S. We've used this many times to validate our early budgets to owners, and it's a key thing that helps us understand the cost of the project and gives you more accuracy and cost certainty going forward. I've utilized 5 projects that we've done across the U.S. The one on the top is one in New York for CUNY, and this is by interiors right now. And of course, the colors represent the same type of things. The dark blue is the interior construction, and that light blue is the HVAC. I mentioned that because you can see all across these 5 projects, the light blue is in the same alignment as opposed to the other projects, meaning that their costs are the bigger costs of the pie than the other pieces. And it's a key indicator for us to understand the cost of a project when we give it to you earlier on. The box on here that's outlined on $620, that is the average cost of the space fit out in each one of these projects. Also on the left, you can see a location adjustment by say, California versus New York. And to the right of that is the escalation factor to escalate to its current quarter. This number, by the way, is escalated to today's quarter, and I'll get into that more later. On the bottom here, there's percentages shown here for program. Program's another thing that we look at very closely, as some programs cost more than others, so you understand where your money is going. On the dark blue, that's the wet labs versus the green, which is the dry labs. When you have more of the wet labs involved with the building, you know it's going to cost you more because those particular spaces cost more money. Next slide. So I mentioned earlier about escalation. So Skanska has taken upon itself to provide a market trends report on a quarterly basis. We started this over 2 years ago now, since COVID, and it provides us and our clients with cost trends, oversized trends about what's going on in the marketplace today. As we all know, the market's going crazy, absolutely nuts in terms of cost. And it is challenging to stay ahead of that and also to inform our clients and educate them what's going on in the marketplace as well. On the fourth quarter of 21, when this is published, building cost index year over year is up 13% over the previous year. That's a lot of money in a year. And the forecast at the time of this published report was that the escalation was forecasted to be between 4 and 8%. Well, that's changed since the first of the year. As we all know, that there's things happening in the world that impact us, like Ukraine and Russia. So for the first 3 months, the escalation rate has gone to 3.74%. That's in the first 3 months. That's more than the 35-year history of tracking escalation on an annualized basis. That's a hefty number. And I would say that today, I would say to our clients or future clients that if you're looking to build something, I would use 8% right now as escalation for all of 22 and maybe into 23. But again, we don't know what's going to happen in 3 months here or 4 months. It might go the other way. But for now, just to play it safe, I would say 8% is good. Next slide. Also, as part of our market trends report, we also keying on supply chains. So as we all know, the supply chain has been really bad in the last year, although the signs at the end of 21 are showing that there's easing of lead times of a lot of major equipment, like air handlers. In the past, they were forecasting 32 to 35 weeks for being an air handler. Now it's coming back down to almost reality. It's around 20 to 24 weeks. So some things like that are easing off. Transportation logistics are also starting to show signs of recovery. So again, the forecast is saying that the supply chains will start to ease and improve once the inventories start to catch up and the transportation logistics start to ease as well. Last slide, please. Not the last slide. So the other things we'd like to track is the Architecture Billings Index. You may wonder why, because as constructors, we have to know where the marketplace is going in the next 6 to 12 months. And Architecture Billing Index helps us with that, as it shows or indicates where the architects are billing for their work for the year. So for here, this billing index currently shows that for 21, the index was up slightly. In the Northeast area, it was just level off right now. But we think that the billings index will improve in the next 3 to 6 months in the New York metro area. I know that we're busy. I know a lot of my competitors are as well. But it's a good key indicator to watch the marketplace. Next slide, please. So I said a lot of things here today, and some of the things not so great, but just the reality of the world today is the inflation. So a couple of recommendations I would give to you if you're planning a fit out of a lab space or building a new building. Try to identify your long lead items and consider strategic early procurement packages, meaning that try to get your air handlers up front quickly, and also your lab equipment and casework as well. Get those out front so you can get ahead of the curve of the supply chain and also the costs. Next one is select the CM with MEP expertise in-house, like Skanska. I know it's a shameless plug, but that's who we are. And we have other people out there who are just as good as us, but I like to think we're better. But you need to have a CM that has this MEP expertise in-house to help you through the pre-construction phase of your project and into the construction phase to give you cost certainty as well as schedule certainty. And again, I'll say it again, budget 8 to 10% escalation annually for your project if you're thinking of anything going forward this year. And lastly, the Architectural Billing Index to watch for continued growth. Now, I would be happy to give you an in-depth presentation on both our Skanska metrics and market trends reports if you so desire. Just let us know. We can bring something further down the road. For now, that's it. Next speaker.
Thank you, John. Yeah, we will be talking more about this, and one of the pieces of the story that, and I'm just going to chat while you bring your slides up, Anthony. So I'm going to introduce you in a moment if you want to start getting ready and share your screen. Have we got Anthony as a co-host?
Let's see. I can share screen now. My slides are ready to go.
There we go. I was just going to say, and I'd like to, when we get to our discussion time, I'll ask you more about this, John, is that the different kinds of programs and that balance of wet labs and the rest of the world, because I think that's really key. So I want to welcome Anthony Caius from Synthego, and one of the key elements of growing companies now, of course, is that they are decentralized. Not everything happens inside the lab anymore. And we're going to hear about a really interesting resource for CRISPR work. Anthony Caius.
Yep. Hello, thank you for the introduction. Thank you for inviting me to come along in this. Yeah, so my name is Anthony. I am the New York State rep for Synthego. And what we do at heart is scaled CRISPR editing. And in terms of how it works in the future in the lab, that's what I'm going to go through in the next 10 minutes that I have and the bunch of slides that I have here, and how we see the future of the lab at Synthego and how it can play in with some of these expanding companies, both pharma and higher education. So in case any questions come up, this is my direct information. I'll leave it on for a second to for anyone to take screen caps for that. In terms of lab work itself, we actually are derived from SpaceX, oddly enough. So even though we're a CRISPR company, we were started by SpaceX engineers, the Dabrowski brothers. So we have taken those engineering principles and brought them to a biology background with the aim of making genome engineering accessible to all, at no matter what the scale, no matter what the size, no matter how big or small any company is. And how we do that is based off of a machine learning artificial intelligence algorithm that's constantly increasing its scope, increasing its abilities, increasing its speed. In the past 20 years of my career, in the first 10 being as a scientist itself at eventually Eli Lilly, and the last 10 being on the sales side, what we've seen is that the cost of the human genome, has decreased. And as it's decreased from the beginning of $100,000 per genome down to about $1,000 per genome, more people have had access to it, more people have more data, basically. But with all that data doesn't necessarily result in more biologics into the market. One of the side effects is that a lot of data also means a lot of noise, right? And what we're trying to do here is trying to help you target through the noise to get to what you really want to get through. And for us, that comes via CRISPR. And for those people who haven't worked with CRISPR before, who don't have any experience with it, what it is precision genome editing, for lack of a better term. It can be used in various locations. It was actually discovered in bacteria. The Nobel Prize winners in chemistry from 2020, Dr. Dowden and Dr. Charpentier, won it for discovering what bacteria do to prevent reinfection of itself. And what we've done here is, in CRISPR in general, not we at Synthego, but we in the scientific community, has used CRISPR to edit specific cells and specific genomes in cells, whether to knock out cell expression or express specific variants of other genes, like some genetic variants in disease states. So basically reproducing clinical data in more of a lab-controlled environment. But this, as you can imagine, is difficult to do manually. So you need to design everything from the RNAs to using the enzyme to determining the break point that the CRISPR system will break. That takes a lot of work and a lot of bandwidth and a lot of thought. And in fact, I kind of show it in three numbers: 43, 7, and 100. Right, and this is what I tell a lot of people to start with, and what this means. Why are these three numbers important? Because when you do this by hand, most of the time, 43% is on the average editing efficiency. If 3 out of every 5 cells that you try and edit are not actually edited, how good is your data, right? So the 7 comes in because it takes people usually 7 attempts before they get a CRISPR model, and up to 100 man hours to create that model, which is, assuming you're working on it, maybe 2 to 3 hours a day, this can take weeks upon weeks upon weeks for probable failure, right? So what this means in terms of the science is that only about 15% of people who manually work with CRISPR actually are satisfied with the results that they get from CRISPR. Expand on that to where data is headed and where we've seen data be headed. Just in the 15 months that I've worked at Synthego, what we found is that more and more researchers, no matter the institution, are coming from a larger data set. I've nicknamed it a GWAS, or a genome-wide association study, which it can also be anything from clinical patient data to electronic patient data to algorithmic-based residues on amino acids that people are looking at. What people do is they take that study and they get a whole bunch of hits out of it. And in the traditional scientific world, the one where I came from, you take that entire list and you filter that down. You drill that down from 100 down to 5. I come in at this point to help model your edits of interest, and hopefully one of them works. This is roughly a 6-month process. If one of them doesn't work, you got to go back to the beginning and do another 5. My postulation is that you're not really looking at your data. You're looking at the filter that you use, your ranking system, to get from 100 to 5. In a perfect world, what we would be doing is looking at all 100 at once. This is your data set. Right. This is where the lab of the future should be. You should be able to look at all 100 at the same time. The reason we can't do it is because of the lab of the present, which involves these things. Pipettes, all scientists use them. Most scientists hate them. I hated mine when I was a scientist, mainly because different pipettes do different things, and different people using different pipettes give different results. The way I like to describe it is, I made my daughter a sandwich for lunch today. I will make her the same sandwich for lunch tomorrow. It's going to be a different sandwich, even though it's the same person making the same sandwich with the same ingredients, because tomorrow is different than today. Which brings us to what Synthego is. In a nutshell, what Synthego is a high throughput, scalable, automated CRISPR factory, right? And the more data that we do, the more experiments that we run, the more data that we get, the better we can update our model so that we can bring those learnings to the next round of experiments that we do. It is a closed loop system, and the more that we do, the better that we get at it. All right, so in essence, what the lab of the future involves is a high throughput, scalable, automated data factory. And what that means for Synthego is that it can be taken in 2 separate ways. That can be taken in the actual RNA that we use to cut the genomes, which is what we call HALO, or we can take that into a cell engineering process, which we call ECLIPSE. In HALO, we can take that in terms of 100 to 50,000 separate guides against an entire genome that we want to cut to look for specific hits, or we can scale that in terms of manufacturing. So if we have one hit and we need to make tons and tons and tons of it, we can scale it that way too. In terms of cells, or actually, let me go. Right, there we go. That's that. I forgot to change the slide. So in terms of the guides, we can take it into multiple plates, or we can take it into large level manufacturing for something like a clinical trial. All done via the same closed loop system that we have. In terms of the cells, we can either take that in terms of multiple edits in the same cell line, or multiple cells with the same edit. What we've done this for recently with some of the researchers that we've worked with has been in the IPS level, where induced pluripotent stem cells, where researchers are trying to recreate data that they've seen whether it be a cardiomyocyte or a brain cell or something like that, these are very, very difficult things to handle. If we can make the research fit the parameters of our factory, we can tell you pretty much what's going to happen and how long it's going to take. So the entire process from growing the cells to transfecting the cells to validating the cells to genotyping the cells, all of it is done via an automated process that limits the variability of someone's hands, quite honestly. So in terms of where this fits in large scale research, right, pretty much all you would have to do is tell us what you're looking for. Are you looking for a target that you want to ID? We can take that through the Halo system. If you have a list of targets that you want to recreate in tissue, or in cells, I should say, we can take that through the Eclipse system. In essence, what we're trying to do in terms of the 50,000 foot view is limit the time to get to clinic. So in some of the studies that we've run, there are other studies that involving Alzheimer's. I think this was a COVID study that we have. We were able to create almost 300 models in less than 16 weeks, all at the same time, all based on our own automated system, which means that frees up your scientist time to think about the actual project and not think about doing the project. For the scientists still at there, the way I like to describe it is when I met a grad student who graduated in 3 fewer years than I did, when I found out that she didn't have to pour acrylamide gels anymore, I was kind of dumbfounded and flabbergasted, and I realized 2 years later that she was right and I was wrong, because she spent her time writing papers and getting reviews. I spent my time looking for dark rooms and looking for reagents, right? In essence, she commoditized her data. And in essence, that's what we are trying to do for CRISPR. We are trying to commoditize CRISPR so that you can spend your time thinking about the data and not how to derive the data, and getting from model to clinic in less than a year. So if you guys want to know more about it and where we're headed from this, these are our websites. These are the resources that we have. We always have things coming up in the hopper. If you are interested in any of it, just let me know.
Well, thank you. I'm going to take host prerogative and ask you a couple of questions, Anthony.
Yeah, absolutely.
Absolutely. And I have a couple of questions for John, and then we'll invite everybody on the call to participate as well. We'll be calling on you and invite you to introduce yourself, make any comments, questions. This is our discussion time now, and we have plenty of time. We do record the session, and we probably our editor just went off and got himself a new job, and we're very proud of him, but we will not have deep editing capabilities. And I only say that because we want to put up the video on YouTube, and we will include the roundtable this time. So just so you know that, I don't think anybody's ever giving away trade secrets in these discussions, but we usually don't put up the discussion time so you can really feel free. But here we go. So I just, it's really quite exciting. I've been doing CRISPR conferences with different organizations for many years, from Boston to RTP, and this emergence is fantastic. Can you get a little bit more specific? You don't have to name a company or a particular researcher unless you want to, but can you share some of the stories about how the work that you're doing? I mean, that's a pretty big claim, from model to clinic in less than a year. So you got some examples of how that's worked? So
the largest scale example that I think we have, and this is public, Bill Scarnes from the NIH has an Alzheimer's study called the INDI. And he came to us looking for 5 edits, because most scientists, like by default, think in terms of 5 or 3 to 5. We were looking at his data set, and that part I don't remember offhand how he came from. Like that GWAS slide that I showed was kind of based off of his study. He had roughly 300 separate variants of the same gene that he had seen in clinical data, and he wanted, in a perfect world, you'd model all of them, right? Not just the 5 that jump out at you for one reason or another, because you never quite know which of those edits are actually going to be the one. Like, everything has a little bit of bias because humans have bias. Even the algorithms that we make have a little bit of bias on them, and the only way to fix that bias is to look at everything at once. So we were able to load the system to create every single model that he ever wanted in less than 16 weeks. So showing that slide where it normally takes someone roughly 10 weeks to create a single model that 6 out of 7 times would fail versus 266 models in less than 4 months, right? Which he could then, because they were IPS cells, differentiate them into neurons, into glia, into whatever model he really wanted to. This is the exact example of he's using his time to think about his data instead of using his time how to come up with his data.
I'm reminded of the Luddites, and it's a really, and I won't spin multiple examples I'm thinking of in my own experience, but that this is pretty revolutionary in what it can actually do. And when was your lab established? What was the factory established?
2002, I want to say. Yeah, everything is done. All the edits are done in our cell factory on the West Coast, right outside of San Francisco.
That's great. And so we wanted to include you in the conversation about the Lab of the Future. We've started an interview process with potential users of a facility that we have in mind for Westchester that would be a mix of education, place for growing companies, and opportunities for real great research and entrepreneurship, but also study throughout the process. And so clearly, not everybody has everything under their own roof. And so the way that you can expedite this process, I think bodes well for wet labs. That there's I like to say, as hard as it is to become a scientist, to get a PhD, it's even harder to stay at the bench, right? And so a lot of the things we think about are how can we support this group, right, who are really the people making the discoveries for tomorrow, right? I think what's interesting about the role that you have is that obviously you can communicate with a lot of different centers and have impact on a lot of different research. So I think there's a wonderful matrix of scientists really working together across nonprofit or academics, government sites, and industry, let's say. So I think that's a really interesting example. Just one more thought is, can you describe to us sort of what's the minimal lab that's going to be able to work with you? Everybody's got kind of an entry point, and we are talking to people that are in, it's college environments that really could benefit from interacting with you.
It's funny. It's a good question, because I have actually worked with virtual companies in the past. At this company even, where the entire company is decentralized. It's incorporated in New York, but the scientist that I talked to was in Oregon, and they're sending their cell models, I want to say, to Wisconsin, right? So there is no lab space in particular. They have one lab to create something very, very small, but the aim for them is, once again, to scale everything up. So we would automate the process of editing the cells that they send us, right? Which we would send then to another state for them to turn those edited cells into whatever target tissue they want, whether it be fibroblasts or this case, smooth muscle tissue. So I would almost argue that in some cases, the minimal lab space could theoretically be 0.
Okay.
At least for my part of the process.
Mm-hmm.
Now, downstream for this, obviously, there's going to be other things involved. But You know,
Well, and the model you described, we know is going on in the Hudson Valley. You know, Michael and I have had conversations with, and Denise, with multiple CEOs that are working out of their basement, and the research is in Canada, and the front office is in Boston or whatever. So I think I like this picture. I appreciate you filling it in. Thanks so much, Anthony. John, the question I had for you, John, is in one of your slides, you talked about the programs, different kinds of programs that people were working on, and I'd love to hear you talk about what you're seeing and how that may have shifted over your career is fine, but even just currently. And you talk a little bit about the program areas, maybe the ones that are forward-looking, and then how you see that may have been changing.
I can comment on the jobs that we're currently working on. So, we see a lot of wet labs, of course, along with the full service of a lab, which is like vivariums and things of that nature. And there's all sorts of different lab types, like BSL-1 through BSL-4. BSL-4 being the highest containment and the most expensive, of course. When you talk about vivariums, they're very expensive. So, all these things need a lot of air changing, special systems, watering systems, all that stuff. So what I see thus far is that a lot of institutions and some private ones are building more lab space because they need more scientists to go after the things like that are very important today, like COVID and everything else, and things like Anthony was talking about. That's very interesting. So in the spaces that we've done so far, like the public health lab, that has numerous lab space as well as office space, and then one floor is dedicated solely to BSL-3 type labs. So very, very, very unusual.
I don't really like to use the word blessings and COVID in the same breath, let alone the same day. However, there is a way in which this emergency has accelerated innovation and collaboration, in a very positive way. We had a program about a year ago on clinical research in general, and we had an expert talking about how the guidance for using telehealth in clinical research, thank you there, it's in your moment, was written like 15 years ago. And everybody was too afraid to include telehealth in clinical research because, of course, you have to go all the way through with the FDA, and they might send you back to get the broomstick of the Wicked Witch of the West, and nobody wanted to take that risk, right? And so I think that this appreciation that, nobody should have to work from their home all the time. However, the whole balance of where you spend your time, it can be very different than it was. And I don't really think we're going to have a post-COVID era because it's going to still be around, but that we'll really be continually developing, multiple and decentralized programs.
Sure. Even in construction, there is what we call hybrid schedules for people like me and other back of house functions in the construction company, whereas we can have flexible schedules. Either work virtually from home or you come to the office several times a week, which is what I do. Unfortunately, for the guys who are in the field building the jobs, they have to be there every day. So there's a little bit of a mix of differences of who works where, who does what. But we're working through it.
We all are.